There we go. And we're capturing audio today, so that's good. All right, so glycolysis is overall the equation that it describes the process is, one molecule of glucose, 2 of ADP. I'm trying to balance things out here. So 2 molecules of inorganic phosphate. Remember, that's just one of the conjugate bases of phosphoric acid. 2 molecules of NAD plus and water just for a proton to have somewhere to go. And in the end, we're going to have, after glycolysis, 2 molecules of pyruvate. Pyruvate is this 3-carbon carboxylic acid, an alpha keto acid. 2 of ATP, 2 NADH, and 2 H3O plus. All right, glucose is a 6-carbon sugar, so we're going to break it into 2 pieces, 2 of the same thing. We're going to get 2 molecules of ATP out and 2 molecules of NADH. ATP can be used for, whoa, sorry about that. The aspect ratio of this bottle is really kind of inconvenient, tips over. I have a sticker on this bottle that has Darth Vader's face on it and the word Nebraska. It's inexplicable. I don't know. Somebody gave it to me. They got it from Trader Joe's on May the 4th, be with you, weeks ago. And we do not know what Vader and Nebraska and Trader Joe's have to do with each other. Just, yeah. All right. So, yeah, it's the state of Nebraska trying to get on board the May the 4th excitement. All right, so there are 9 or 10 reactions in glycolysis, and some of them involve what's called the investment phase, where we're actually going to be consuming molecules of ATP in order to get the glucose molecule rearranged to the point where we can break it apart into different pieces. So what I'll show you now is the investment phase of glycolysis. We'll draw some of these mechanisms. Others we'll simply talk through because you've seen similar mechanisms before. I want you to know the molecules, their names, and also the names of the enzymes. So glucose is, of course, easy. You've done that before. 6-carbon sugar. And we're going to consume 1 molecule of ATP, and we'll get 1 molecule of ADP out. And we're going to convert the phospho and hydride, and I'm not going to draw the structure of ATP because we've done that recently, so you can go back and look at that. We're going to convert the phospho and hydride of ATP into the phosphoester that's more stable. I'm using the squiggly line at the anomeric carbon to indicate that we've got a mixture of alpha and beta anomers, and we don't really care. The product is called glucose 6-phosphate. It's the carbon 6 hydroxyl group that gets modified. The enzyme that does this is called hexokinase. Kinase is a word we use for enzymes that transfer phosphate groups from ATP. You will see that again and again in biochemistry. Yeah?
I know for the MCAT we have to know certain enzyme, aldolase, hexokinase, and one other their mechanisms. For this class, do you care if we know the mechanisms?
Oh yes, I do care that you know the mechanisms. Yep. So this hexokinase catalyzes reactions where you transfer phosphate groups from ATP. This is downhill in energy, as you might expect, because you're replacing a weaker phosphoanhydride bond with a stronger phosphoester bond. So this is downhill in energy. You don't need to know by how much, but I'm putting a number here just for context, and this is essentially irreversible. So once a molecule of glucose embarks down this first step, there's no going back from glucose 6-phosphate to just regular glucose. This is a commitment step. And most of the steps in glycolysis that are irreversible are also highly regulated. We won't talk about that in OChem. That's a subject for biochemistry, about how enzymatic steps are regulated. But it makes sense. You don't want to commit to something if, for example, you don't need to metabolize glucose at the moment. And so hexokinase is one of the regulated enzyme steps. The mechanism of this step is basically an SN2 style mechanism like we've seen before. If you want to draw it, you could just abbreviate the alcohol of glucose and then draw the phosphoanhydride of ATP. Oops. Basically, we'll have some enzyme remove the proton from the hydroxyl group of the sugar, and we'll attack the gamma, remember alpha, beta, gamma phosphate groups of ATP, and we will break this weak phosphoanhydride bond. We're going to see a number of steps that are like that. We probably won't draw that mechanistic step again, simply because it's going to be very similar. Okay, questions about that step? Kinases transfer phosphate groups. Hexokinase means it's a kinase for a hexose, which is a 6-carbon sugar. All right, step 2 is isomerization. So we're going to take glucose 6-phosphate and we're going to isomerize it to fructose 6-phosphate. And I'm abbreviating the phosphate group like that. All right. Interestingly, the enzyme that does this is called glucose 6-phosphate isomerase. What does that enzyme do? Well, it makes an isomer. And I'll show you what that isomer looks like. It's going to have the same stereochemistry at carbons 4, 5, and 6. Let's see, sorry. 1, 2, 3, 4, and 5. It's going to have the same stereochemistry at carbons 3, 4, and 5, but we're going to end up with a 5-membered ring instead of a 6-membered ring. So notice how the hydroxy methyl group on carbon 5 is pointing upward in the original glucose 6-phosphate starting material. Still up in our product. The OH on carbon 4 is still down. The OH on carbon 3 is still up. One of the things you'll notice, though, is we've moved the hemiacetal carbon over. The hemiacetal carbon used to be carbon 1. And now it's carbon 2. Now, remember how we said that with the hemiacetal, you can tell whether that carbon used to be an aldehyde or a ketone based on what's attached to it, right? For carbon 1, you're attached to 2 oxygens, a carbon, and a hydrogen. meaning carbon 1 used to be an aldehyde. How about carbon 2? 2 oxygens, 2 carbons, so carbon 2 is a what? Ketone, right? So we've switched from an aldehyde to a ketone. We've basically moved everything 1 carbon over. Mechanism here is basically just involves proton transfer. So I'm not going to show the mechanism of opening of a hemiacetal because you can do that or should be able to do that. So if we open up the hemiacetal, here's what we've got. Carbon 1 is an aldehyde. I'm also not going to show closing of the hemiacetal ring or forming of the hemiacetal ring for fructose because again, you ought to be able to do that based on things that we've done before. All right. Anything you want to ask about opening or closing of those rings? Now, how are we going to get from having a ketone on carbon 1 and an OH group on carbon 2 to having an OH group on carbon 1 and a ketone on carbon 2? It just, what was that? Sorry, I was going to say something. Tautomer, okay. So what's the tautomer of an aldehyde? We got a word for it. It was a enol, right? And you get that by removing an alpha proton first. Or rather, this is one of the ways you can do it. Remove the alpha proton first to get an enolate. Okay. And then I can protonate that enolate on the carbonyl carbon. What's the base and what's the acid? Those are side chain or backbone groups on the enzyme. Once I've done that, let's see what the intermediate looks like. All right, this is called an ene, and we could just call it an enol because we have an ene here and an ol there, but sort of on accident, it's an ene diol. And this is the key intermediate because the ene diol looks like an enol in this direction, but then it also looks like an enol in that direction. So we've got an intermediate where carbon 1 used to be the carbonyl carbon and carbon 2 used to be the alpha carbon. And we could still go backwards, but do you see how, because of the presence of the OH group on the alpha carbon or on carbon 2, this could also be a carbonyl carbon and that could be an alpha carbon. Okay, so that ene diol intermediate is key because it allows us in the next step to remove a proton from the orange oxygen, and that generates another enolate, only this time the alpha carbon of the enolate is carbon 1. Oops. So now if we kick these electrons down and protonate on orange alpha carbon, that gets us to the product. And the product is called fructose 6-phosphate. The only difference between glucose and fructose is a ketone sugar, glucose is an aldehyde sugar. They're isomers of each other, and you get from one to the other through 2 tautomerization steps. So this series is the first tautomerization, and then this series is the second tautomerization. And the key is this ene diol intermediate. Now you might expect that this reaction is reversible, and it is. There's not a huge free energy change, so it's easy to go backwards versus forwards. All right, questions about that reaction? Sure.
Is it reasonable to put the ketone on any of the OH, or like you just keep moving down, or is it not going to do that?
Right, let's talk about that. So could you do subsequent tautomerization reactions with, in general, if you have an alpha hydroxy ketone, you should be able to access an ene diol intermediate. Now let's talk about why nature wouldn't want to do that. Remember that overall, we're going to want to split glucose into 2 3-carbon pieces, okay? So I want you to look at, in particular, the open chain forms here of glucose 6-phosphate and fructose 6-phosphate, respectively. And I want you to think of reactions, well, let me say this. Do you see an aldol product embedded somewhere in either glucose or fructose? Remember, just as a hint, the product of an aldol reaction is, not a hint, a question. What is the product of an aldol reaction? Beta hydroxy carbonyl compound. Is there one in glucose? Is glucose a beta hydroxy carbonyl compound? Sure, it's carbonyl carbon, alpha carbon, beta carbon. How about fructose? Is fructose a beta hydroxy carbonyl compound? Sure. Now, when we do a retroaldol reaction, when we do a retroaldol reaction, the bond that we would have made during the aldol, and the bond that you can break in the retroaldol, is the bond between alpha and beta carbons. If you want to split glucose up into 2 3-carbon pieces, What's via a retroaldol reaction, what's your best bet, glucose or fructose? Fructose, right? You see the bonds in the wrong place for glucose. In terms of chemical strategy, that's why nature's moving the ketone over, because then the beta hydroxycarbonyl compound, the bond between alpha and beta carbons, is in the right spot, so that when you do the retroaldol reaction, you get 2 3-carbon pieces. It's not to say that nature couldn't have dealt with a 2-carbon piece and a 4-carbon piece. It's just more efficient to split a 6-carbon sugar into 2 3-carbon pieces. Requires less work down the way. So yes, to answer your question, you could have gone on and do further isomerization steps, but that would get you further away from having the beta hydroxycarbonyl compound in the right place. All right. Follow-up questions about that? Yeah.
What was the name of the molecule right before we got to the ketone?
This is an enediol. This one? Oh, it's the enolate of the enediol.
Okay, so then what was the name of the
Open chain form fructose 6-phosphate. Yep. All right. So, the third step, now that we sort of have an idea of what's going to happen here, moving forward, we're going to know ultimately we're going to want to break this bond between alpha and beta carbons. Before we do that, we're going to need to put another phosphate group on this molecule. We're actually going to put a phosphate on the carbon 1 hydroxy group. That may not seem like an obvious step at this point, but you will see later how doing this makes it so when we do the retroaldol reaction, we actually get 2 almost identical pieces that are relatively easy to interchange. So, fructose 6-phosphate. This is another irreversible step. It's going to consume ATP and give us ADP out, and we're going to make a phosphoester. And the new phosphoester is going to involve the C1 hydroxyl group. The mechanism is going to be SN2-like. Same thing we saw previously up here. Remove a proton from the OH group, negatively charged oxygen attacks the gamma phosphate of ATP, breaking a phosphoanhydride bond and replacing it with a stronger phosphoester bond. Oops, I guess I can draw it out. Here's the new phosphoester that we made. The enzyme that does this is a kinase because it transfers a phosphate group. It's called phosphofructokinase because it adds a phosphate group to fructose 6-phosphate. And this is also irreversible. It's downhill in energy, not by quite as much as the first step was, but still by a number that's big enough that the equilibrium constant should be something like 1,000 to 10,000. And the product is called fructose 1,6-bisphosphate. Bis for 2. Okay, 2 phosphate groups. Yeah.
Is there any reason that oxygen attacks the phosphate versus the other?
Versus the others?
Yeah.
Yeah, so you've got one, two, three, four OH groups, and only one of them does the attacking. There's two reasons why you might suspect carbon 1 would get modified instead of the others. First, this is a primary hydroxyl group and is therefore less sterically hindered, so there's that. The other thing is that you're going to see in just a minute when we do the retroaldol reaction, we need the phosphate in this location chemically, or in terms of our strategy to get sort of two similar pieces. Having it in the other locations doesn't help. So the other thing that perhaps controls regioselectivity is the enzyme. And that's kind of a hand-wavy explanation, but yeah, that's what we got. Okay, what else? Yeah.
This is a super silly question, but is there a reason why it's bisphosphate, not diphosphate?
Bis versus di, there's probably a rule for that, I don't know. Yep, no idea. Okay, others? Okay, so we're still in the investment phase. Step 4 is your break apart into two equal portions. That's a step catalyzed by an enzyme called aldolase. And step 4 is the retroaldol reaction. So you've been preparing for this for like weeks, learning about the aldol reaction, and this is the reason why. We're going to take fructose 1,6-bisphosphate and told you the enzyme is aldolase, and that's going to convert trying to decide how to draw this. It's going to convert our 6-carbon sugar into one 3-carbon piece called glyceraldehyde 3-phosphate. And the other piece, and this will have been carbons 1, 2, and 3. Sorry, wrong about that. Totally wrong about that. That will have been carbons 4, 5, and 6 of the sugar. And then the other piece we will get out is this one, carbons 1, used to be carbons 1, 2, and 3, and this is called dihydroxy acetone phosphate. 2 3-carbon molecules. All right. Notice what the relationship is between glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. One's an aldehyde, one's a ketone, but glyceraldehyde 3-phosphate is to dihydroxyacetone phosphate what glucose is to fructose. They're isomers that differ, right? In glucose, we had an aldehyde on carbon 1 and an OH on carbon 2. Whereas in fructose, we had a ketone on carbon 2 and an OH on carbon 1. We've got a similar situation with glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. All right, the key here is, again, just to recognize that, and I'm not going to show you opening of a cyclic hemiacetal because I believe you can do that on your own, based on what you've seen so far. Oops, whoa. I want to do that. How do I stop it? There we go. so here's the open chain form. Again, I'm just going to highlight that this is, whoa, that's not an open chain form. There we go. This is our carbonyl carbon, our alpha carbon, our beta carbon. This is the bond between alpha and beta carbons. The retroaldol reaction involves removing a proton from the beta hydroxy group to generate a negatively charged oxygen. And I'm going to show in the same step those electrons kicking down to kick off the enolate. And that's the retroaldol step. I'm going to keep drawing things as close to, in an arrangement that's close to how they looked in the starting material. All right, so there's my new aldehyde involving carbons 4, 5, and 6 of the starting material. Over here now, I have my enolate. Which I can pick up a proton from some group in the enzyme. We don't really care what it is at this point, and those are my two products. Okay, retroaldol reaction. Questions you want to ask about that? Yeah.
So can these two products form, kind of switch back and forth like glucose and fructose could?
Yes, can these interchange the way glucose and fructose can? Yes, and that's the beauty of this, because the next step is going to be to convert the half of our material that's dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Yep, so at that point we now have taken a 6-carbon sugar and converted both pieces of it into the same thing. This is the efficiency that nature builds in. All right, other questions? All right. This aldolase step is reversible. I've shown you a mechanism that works, and I think it's generally the mechanism that applies in bacteria. In eukaryotic cells, that's you and me, the enzyme actually forms an imine with the ketone first before it does this chemistry. We don't need to show that happening, but if you learn about that later on in biochemistry, you'll know a little bit about what's going on. Okay, so step 5 is going to be isomerization of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, and this step is going to be reversible as well. That gets all of our material from glucose into one pattern of connectivity. From this point on, after this step, everything else we do in glycolysis, we're going to do twice. And again, there's a big sort of efficiency gain there. The enzyme that catalyzes this one is called triose phosphate isomerase. And, or TIM for short. Triose phosphate isomerase is one of the enzymes in nature that is perfect, or as perfect as nature can get. This is not anything to worry about or memorize, but if you think about how fast reactions happen, there's a speed limit to how fast reactions can happen in water, and that speed limit is diffusion. That is, a reaction can't happen any faster than a molecule can swim through water. And the rate of swimming through water is about 10 to the 8 is it molar per second or is it per molar per second? I forget. Anyway, yeah, so there's more to say about that, but I'm not prepared to talk about it. I was gonna try to make some connection to the gospel there, but anyway, I got nothing, so we'll just move on. Sorry about that.
So you think that trios that tin can move fast?
No, it's that the reaction is efficient enough that improving the enzyme could not make the reaction go faster because you're already as fast as diffusion. So basically the enzyme becomes so good that it's no longer the rate determining step in the process. The rate determining step is the molecule even finding the enzyme in the first place. Okay, so this is just going to be the mechanism here I'm not going to draw because you've seen it already. It's going to be 2 tautomerization steps via an ene diol intermediate. I can draw the ene diol for you if you want to help you a little bit along the way. You should make sure you can draw this mechanism, but it's not hard based on what I showed you before for fructose. All right. So that concludes the investment phase, right? You've got add a phosphate group to carbon 6, that commits glucose to the process. Isomerize via an ene diol intermediate to get to fructose. That positions the ketone relative to the beta hydroxy group so that we can, in the third step, so that we can later on in the fourth step do the retroaldol reaction. Third step, we add another phosphate group to carbon 1. You can see now that the reason we did that was so that after the retroaldol reaction, we had 2 pieces that resembled each other quite closely and are interchangeable only through, simply through 2 tautomerization steps. All right, anything you want to ask about that? All right, let's recoup our investment, get a little bit of a payoff. From this point on, I'm not, everything happens twice. Two times steps 5 through 10, 6 through 10 rather, whatever. So step 6 is one of the payoff steps. We're going to take glyceraldehyde 3-phosphate and we're going to oxidize it. So in this reaction, you need glyceraldehyde 3-phosphate as your starting material. And we're actually going to need inorganic phosphate, or PI. And we're going to need NAD+. And the product is the carboxylic acid version. Well, sorry, got ahead of myself. So we oxidize something that used to be an aldehyde up to a functional group that looks kind of like an ester. It's actually a mixed phosphoanhydride. Here's our aldehyde carbon bonded to twice to a heteroatom. Now here's your, oops. it's at the carboxylic acid level of oxidation. You can tell it was an oxidation step too, because we went from NAD plus to NADH. All right. I call this a payoff phase because we got NADH out, and you can use this if you take it to the electron transport chain in mitochondria. You can use it to generate molecules of ATP. The enzyme that does this is called, because this is an oxidation reaction, the enzyme is called a dehydrogenase. Glyceraldehyde. Ah. 3-phosphate dehydrogenase. Or GAPDH, some people will abbreviate it as. I just put that there because if you are ever working in a biochemistry or cell bio lab and you're doing things called Western blots, sometimes you will need to demonstrate that you loaded the same amount of cellular protein in each well of a gel, and to do that you'll assess the amount of glyceraldehyde 3-phosphate dehydrogenase expressed by each cell that you're working with, and assume that that should be constant. Anyway, so you may see that before in papers that you read. You may see that later subsequently in papers that you read. Not a huge deal, so don't worry about it. The product is called 1,3-bisphosphoglycerate. There's that bis, meaning 2, bisphosphoglycerate. And the glycerate is the, it tells you that we're at the oxidation state of a carboxylic acid. All right, let's talk mechanism. Its first inorganic phosphate is going to act as a nucleophile to attack the aldehyde. Nucleophile attacks carbonyl compound to get a tetrahedral intermediate. Let me draw the structure of that tetrahedral intermediate, only I am going to abbreviate a little bit. Notice that in the product we're missing that aldehyde proton. Where does it go? It goes to NAD+. You've seen NAD+ based reductions and NAD+ based oxidations before. We can draw a resonance structure for NAD+, in which the positive charge isn't on the nitrogen anymore, it's on the para-carbon, which tells you that that para-carbon is, just won't worry about what the R group is electrophilic. Electrons kick down and hydride gets delivered directly to that para-carbon. You may draw the mechanism of this reaction either of the two ways I'm going to show you. Either way is equivalent. And that gets rid of the hydride and establishes the mixed phosphoanhydride. And as I said, everything that we're talking about here happens twice, so we just made 2 molecules of NADH. All right, questions there?
So you just drew is NAD+, right?
Right, this is NAD+, and afterwards, after we deliver the hydride, now you've got NADH.
Is there supposed to be an NH2 on this first one as well? On the other side?
Yeah, sorry about that. I was, I guess, in a hurry.
Yeah.
Yep. Okay. All right, let's take a break and we'll pick back up with that in about 10 minutes. All right. Let's go ahead and finish up on glycolysis. If you're following along with what we've accomplished so far, we just barely checked off 2 molecules of NADH, right? The problem is that we are in the hole when it comes to ATP, because not only do we not have 2 molecules of ATP, we already spent 2. First, in going from glucose to glucose 6-phosphate, and second, in making fructose 1,6-bisphosphate. So we need to regenerate ATP, and we're going to need 4, not only to break even, but to come out ahead. But you should, as we go along, you're going to see how nature has adjusted things so that we've got phosphates on good leaving groups and are going to be able to transfer some phosphate groups to ATP. So the last step we just did was oxidation of glyceraldehyde 3-phosphate to 1,6-bisphosphoglycerate, and that's the starting material for our next step. And we're going to focus in on this phosphoanhydride bond that we got by oxidation. Because remember, and this is step 7 of glycolysis, remember that in ATP, the bond between the gamma and beta phosphates is also a phosphoanhydride bond. Right, this bond here is a phosphoanhydride bond in the same sort of way that this bond is a phosphoanhydride bond. We're going to call it, it's not exactly a phosphoanhydride in the sense that what's here is a carboxylic acid instead of a phosphoric acid, but the bonds are similar in strength. Which means we're actually set up, because of the structure of bisphosphoglycerate, because that phosphate group on carbon 1, because that phosphorus is attached to a good leaving group, that is a carboxylic acid, we can now have the beta phosphate group of ADP act as a nucleophile in sort of an SN2 step, attacking that phosphate group, and leaving group leaves. And that's going to generate ATP, and then our leaving group is just going to be phosphoglycerate. Phosphoglycerate. The enzyme that catalyzes this reaction is named for the backwards reaction. It's called phosphoglycerate kinase. the kinase, phosphoglycerate kinase would suggest transfer of a gamma phosphoryl group from ATP to 3-phosphoglycerate to get bisphosphoglycerate. This step is reversible. The change in free energy isn't very high because you're replacing a mixed phosphoanhydride with a similar strength phosphoanhydride bond. But now you begin to see what all the investment is for. It's to get functional groups in an arrangement so that what's attached to a phosphate is itself a good leaving group, so that it's easy to convert ADP back into ATP. All right, questions about that step? We do that twice, so we just paid our debt. We broke even for ATP, but we've got more to do, because remember, we've got a 3-carbon piece here. It needs to be a 3-carbon piece with a ketone on carbon 2. All right. And we still need to regenerate 2 molecules of ATP. All right, step 8 is an isomerase step. The enzyme that catalyzes this is called phosphoglycerate mutase, and it simply moves a phosphate group from carbon 3 of 3-phosphoglycerate. It's going to move that phosphate group from the OH on carbon 3 to the OH on carbon 2. So we're going to have an OH here. The OH on carbon 2 is going to be the one with the phosphate group. And then we've got the carboxylic acid. And the mechanism here is a little bit complicated, so we're not actually going to worry about it. It's not that bad, but the details are just maybe a little bit overwhelming, so we won't worry about it. Basically, it involves a histidine side chain that has a phosphate group attached to it, and you add a phosphate group to this OH group, and then you remove it from that one. All right, step 9 is making Step 9 is going to be an elimination step. The enzyme that catalyzes this is called enolase. And yeah, the mechanism is basically E1CB elimination. We're going to start with 2-phosphoglycerate. And the product is going to have a double bond between carbons 2 and 3. And the product is called phosphoenol pyruvate. So named because if you look at what pyruvate is, that's going to be our long-term product of glycolysis. The enol of pyruvate would look like this. And so phosphoenol pyruvate is the phosphoester of the enol of pyruvate. The mechanism is just basically what you would expect. You remove a proton from the alpha carbon to generate an enolate. And then in a second step, electrons on the alpha carbon kick off OH as a leaving group. It's basically a dehydration reaction. Now, one thing I haven't mentioned is we just deprotonated something that already had 3 negative charges on it. That should seem kind of bad. One thing we're not saying is that phosphate groups in particular are often found in biology associated with divalent cations like magnesium to help neutralize the charge. And so even though we've got a molecule that's highly negatively charged, it's still okay to remove this proton because probably we've got counter ions nearby that are sort of helping neutralize the highly negatively charged environment. Okay. So, last step is going to be the final payoff. It's actually an irreversible step. It's the third of our 3 irreversible steps from glycolysis. You'll take phosphoenol pyruvate. And you'll take the beta phosphate group of ADP. We do an SN2-like attack on that phosphate group, and a leaving group leaves. The reason it can leave is because it's a resonance stabilized anion. I'm not going to draw the structure of ATP. This is called the enol of pyruvate, and it is the less stable tautomer. And I'm not going to show you the mechanism of that tautomerization process because you can draw it. One of the things that provides the driving force for this reaction is that it's downhill in energy to tautomerize from the enol to the ketone. All right. So this is the more stable keto form of pyruvate, except we're having a hard time spelling. All right. So I think there's evidence that probably the pyruvate enol protonates on the oxygen first because it's faster to protonate on the oxygen and then tautomerizes later. It's okay if you want to draw a mechanism where you don't protonate on the oxygen but instead kick electrons down and protonate on the alpha carbon. That would be fine too. All right, that's our 2 new equivalents of ATP. And everything so far was about, after splitting the molecule into 2 equal parts, it was about positioning functional groups and phosphates in locations where the phosphate would be attached to a good leaving group so that you could do SN2 style attack of ADP on a phosphate group. You'll hear biochemists talk about steps, 7 and 10 as examples of what are called substrate level phosphorylation of ADP back to ATP. This is different from the way mitochondria make ATP. This involves a direct attack of ADP on a phosphate that's attached to a leaving group. All right, So there you go. There's glycolysis. Anything you want to ask about that? Yes.
When for step 6 through 10, when you say it happens 2 times, does that mean you have twice the amount of starting materials for each step? And then
Yes, because I made, if you watched in the earlier step, I made 1 molecule of glyceraldehyde 3-phosphate here and a second there.
Okay.
Yep. All right, others? Yeah.
So I'm guessing that the polymerization happens in acidic conditions?
Does it happen under acidic or, you could do it under acidic or basic conditions. Either's fine. You could draw a reasonable mechanism either way. Yep. Remove a proton here, protonate the enolate on the alpha carbon, or alternatively, pi electrons attack a proton, make a new carbon hydrogen bond with the alpha carbon, and then remove this proton. Yep, either way is fine. Yeah.
Will this process also work for other monosaccharides?
Does it work for other monosaccharides? That's a good question. I don't exactly know the answer to, but my guess is no. You've got to have the stereochemistry right. So probably before sending galactose or mannose down this path, probably you've got to isomerize those into glucose first. I could be wrong. Yeah, I don't know. What else? Kind of want to show you how horrible. Oh, Jackbox, we were playing the other night. Adam was cheating actually on Quiplash. He wasn't cheating, but it's easier to draw stuff when you have the Apple Pencil than with your fingers. So, all right. I want to look up. Let's see. Sort of want to give you a sense for how. Yes, that will do. That will do nicely. How complicated all of these reactions are. So you can Google metabolic reactions poster and it will take you here to this website. Roche is a chemical and pharmaceutical company. Let's see if we can find what we've just done, glycolysis. All right. Okay, yeah, so out of that great big, huge, horrible thing of metabolic reactions, we just did that small portion. So let's see. Fructose 1,6-bisphosphate, glyceraldehyde, phosphoglycerate, 3-phosphoglycerate, phosphoenol pyruvate, 2-pyruvate. But the cell is much bigger than that, and there's a whole bunch of other things going on. Fatty acid beta oxidation was there. And we're going to turn our attention soon to this lovely circle, which is called the citric acid cycle. Before we do that, there is a bridge between pyruvate and the citric acid cycle, which we need to talk about. What I'm saying is, if you think things are bad, they get a lot worse. But fortunately, I don't really know anybody in biochemistry that's really interested in you memorizing all of this. So, but if you want to understand all the chemistry that's going on inside the cell, that's kind of an interesting way to summarize it. I'm trying to figure out what the size of this poster would be in order for a human to read it reasonably. I don't know. I think it's big enough that they don't actually print it for you anymore. I think you have to just look at it online. Yeah.
Some nutrition
Okay. So decoration for your house someday on your ceiling. So when you're in bed at night, you're just looking up at that thing and yeah. I prefer Star Wars art myself, but yeah. I had a student once come to my office, look around. Those of you that have been there, look around and say, "So, you like Star Wars, huh?" I'm like, yes. Is that a problem? Note to self, don't insult the professor when you're coming to ask for points, right? This is the thing my children are starting to realize. One of them in particular, the 15-year-old used to. He's much better about it now, but when he wanted something, he would say he wanted something, and then when I was resistant to it, he would start insulting me. I'm like, buddy. And then he doubled down on the insulting. I'm like, this is not, even if you think you're right, this is a bad strategy for getting what you want. So that's all right. So to get to the citric acid cycle, we need to take pyruvate and convert it into acetyl-CoA. Acetyl-CoA, as you might recall, is the endpoint of fatty acid beta oxidation. So, and this is the entry point to the citric acid cycle, which is essential to aerobic respiration or metabolism. There's a multi-enzyme complex that does this reaction. It's called the pyruvate dehydrogenase complex. Dehydrogenase tells you it's going to be an oxidation reaction. And we'll talk about how all this happens. It's kind of complicated. It's also kind of beautiful. We need to lose CO2 in the process. And let me see. And we're going to generate NADH as part of the process. One of our challenges here that we need to solve, and we need to bring in coenzyme A, the thiol. Okay. So look at what's changing here. We need to break a carbon-carbon sigma bond. Right? We want to break this one so that we can get CO2 out. Here was our carbonyl carbon. This is our alpha carbon. We got to oxidize somehow this alpha carbon from being a ketone to being a thioester. We got to get rid of CO2, but all we know about for getting rid of CO2 right now is decarboxylation of a beta keto acid. We've talked before about how it's possible to take a beta keto acid and do a decarboxylation reaction because the resulting leaving group is an enolate, which is resonance stabilized, right? That's decarboxylation of a beta keto acid. The problem with decarboxylation of an alpha keto acid is because if CO2 kicks off, the negatively charged electrons haven't got anywhere to go. They're stuck on that carbonyl carbon, but they're stuck in an sp2 orbital not stabilized by resonance. You just got a carbanion there, and that's bad. So decarboxylation of a beta keto acid is easy. Decarboxylation of an alpha keto acid is not easy and requires cofactors. Okay? But I want you to pay attention to that structural pattern of an alpha keto acid, because what we do here is going to be similar to what we do later in the citric acid cycle for a similar set of molecules. All right, so what happens here happens in 2 or 3 steps, and we'll talk about them. First is what I'm going to call oxidative decarboxylation of pyruvate. All right, and there's some cofactors here that are required. This is pyruvate, and let's see. Cofactor 1 is something called lipoate. This is weird. It's a 5-membered ring containing a disulfide. And then the molecule that has that disulfide in it is called lipoic acid, and it is attached via an amide bond to a lysine side chain. So this is a lysine side chain, alpha, beta, gamma, delta, epsilon. This is lipoic acid, and the business end of lipoic acid is this disulfide. And the enzyme is going to so those are the 2 players, and then there's a cofactor called thiamine pyrophosphate ylide. Or TPP. I'll draw this for you. Oops. I can't remember what vitamin thiamine is. I just remember seeing the word thiamine on like cereal boxes, so presumably we eat it. Thiamine has a 5-membered aromatic ring. Unusual because you got a nitrogen and a sulfur in it. It's called an ylide because thiamine itself is the conjugate acid of this molecule, would have had a proton there. You got a permanently positively charged nitrogen here. If you remove that proton, the molecule can be neutral, at least in this area. And the properties of this thiamine ylide are really kind of interesting. The lone pair here associated with that negative charge is held in an sp2 type orbital. You can draw a resonance structure of thiamine ylide in which the adjacent nitrogen isn't positively charged. So I can move these electrons from the lone pair onto that nitrogen. And then it's as though you have here a carbon that has a positive charge in an empty p orbital, but on the same carbon, a negative charge in an sp2 orbital. In other words, the same carbon is both a nucleophile and an electrophile. At this point, sometimes people drop the charges entirely and they just use that structure. It's called a carbene. Molecules like this that have a nitrogen adjacent to this carbon are sometimes called N-heterocyclic carbenes. I'm going to keep the charges because that helps me remember that this carbon is a nucleophile in an sp2 orbital and an electrophile in a p orbital. That's going to be important for what thymine does, and I am going to show you that mechanism. When we're done with the reaction, the product is going to be CO2, so we're going to lose this CO2 group, and then the remaining carbonyl group is going to end up attached to the sulfur, one of the two sulfurs of lipoic acid. Okay. So how does this happen? I mean, any questions about overall about what's going on? I think one question, go ahead.
Is that lipoic acid the COASH?
No, that's not coenzyme A. It turns out this is going to be consumed and regenerated during this process, so it doesn't show up in the overall equation.
Oh,
Yeah. Yes?
Does that ring break then?
Yes. 5-membered ring breaks. The disulfide is an oxidized form, and here it's going to be reduced. The thing that gets oxidized is the carbonyl carbon of our ketone gets oxidized to a thioester. The thing that gets reduced is the disulfide, which gets reduced to the thiol. All right, other questions? So let's look at mechanism. I'm going to abbreviate the thymine group because the central portion is the portion that participates in the reaction. So here's your alpha keto acid. And here's your thiamine illid. I don't know, there we go. So nucleophile attacks ketone to get a tetrahedral intermediate. I'm going to not show the negatively charged tetrahedral intermediate. I'm going to show it getting protonated in the same step by some acid. It could be a group from the enzyme helping to catalyze the reaction. Okay, so the new bond we made is right here. Remember how I told you previously we want to decarboxylate, but we got a problem because we got a ketone on the alpha carbon, not on the beta carbon. After the thiamine illid attacks, sorry, I guess I was missing a double bond there. let's look at how the structure of this molecule has changed. And in fact, it might be worthwhile, I don't know, redrawing the molecule. And I want you to see if you can see any resemblance between these two molecules. Do you see how now with the addition of that thiamine group, on the beta carbon, I have something that looks like a ketone now. So that thiamine converted my alpha keto carboxylic acid into something that now sort of looks like a beta keto carboxylic acid. And now I'm set up for a decarboxylation reaction because if electrons kick down and CO2 leaves, let's draw what the leaving group is. And let's see whether we think it's stable enough to tolerate leaving. All right, what do you notice about that leaving group? Resonance stabilized, right? It's basically an enolate. We could draw that in a couple of different ways. Ah, sorry. Okay. So the thiamine group that we had attack as a nucleophile, as soon as it's on, acts as a place for the electrons from this bond, the carbonyl carbon to alpha carbon bond, acts as a place for those electrons to go and be stabilized by resonance, right? Any of these, we've basically got something that was neutral, right? The thiamine acts as what we call an electron sink, or a place for the electrons to go. All right, anything you want to ask about that? That's the decarboxylation step. So check, we've got that guy gone. Where does the lipoic acid come in? I'm going to bring that in at this stage. And I'm going to copy this resonance structure of our thiamine sort of leaving group. Negative charge on what used to be our alpha carbon. it's still nucleophilic. It's stabilized by resonance. And it turns out that disulfide bonds are, heteroatom bonds in general are relatively weak. And so we can have this anion attack the disulfide, and a thiolate leaves. We can show it actually picking up a proton on its way out. So what we get after that and I'm going to just use SR at this point. Then a base can remove a proton here. We can kick off thiamine ylide as a leaving group. That regenerates the thiamine ylide cofactor that we started with. And gives us our thioester, which, if you want, we can draw, but I already did above, so we'll just copy and paste. All right. Questions about that mechanism? The purpose of the thiamine is to act for a place for the electrons to go after the decarboxylation. And then the electrons that are sitting on that, what used to be our alpha carbon, act as nucleophiles to attack this disulfide. And that new bond that we just made is still present in our thioester product. That's kind of a lot. That's kind of the worst one. So, moral of the story, decarboxylation of beta keto acids, easy. Decarboxylation of alpha keto acids, you need all this machinery to make it happen. But thiamine pyrophosphate ylide is the key functional group that allows all of that to happen. Oh, all right. Are you at the point where you're like, whatever, I'm just going to have to look at this on my own,
Yeah.
basically? Okay. Go ahead. You're just agreeing, yeah? Okay.
So is the final step just to sub it with
So
seal?
next, the second step of 3 is basically a trans-thioesterification, where you bring in the acetyl-CoA thiol and you swap out the acetyl-CoA thiol for the other thiol. So it's just, I won't draw that mechanism, but that's basically nucleophilic acyl substitution. Trans-thioesterification. Nucleophilic acyl substitution. Okay. That is, I agree, going to take some time looking at it. What's confusing right now, aside from like everything? Can you be specific?
Yes, you know that one step.
That one step? Okay. All right, well, fine. This is that point in 3rd Nephi where you get sent home to think about things instead of sit there and worry about it anymore.
And then we start crying and kill people.
Oh no, you're crying with relief to be leaving me, I'm sure. All right, see you next time.